Next topic is about chemical digestion. Enzymes are proteins. Not all enzymes, most of the enzymes are proteins. Some of the enzymes are RNAs, but again, the majority of enzymes are proteins. Now, the names of the enzymes usually end with ASE. So if you see a term that ends with ASE, there's a good chance that it's some kind of an enzyme. Right? So for example, enzymes that break down proteins are called proteases. So you can see ASE right here, right? And there's a special type of enzyme that digests starch. It's called amylase, and that's because amylose is a type of starch. So amylase specifically targets amylose. I don't know if you remember. Oh no, yeah, we haven't talked about carbohydrates yet. So usually sugars, carbohydrates, sugars are a type of carbohydrates. So they end with the words usually end with OSE. For example, lactose, milk sugar, right? So that's milk sugar, and you can see it ends with OSE. So amylase is the enzyme that specifically breaks down amylose. Amylose is a type of starch. Lipases, so these are going to be enzymes that digest lipids, right? Such as fats, and we have a lot of fats in our diet these days. So we definitely need those enzymes to digest fats. Nucleases, these enzymes would digest the nucleic acids. Right, so just kind of a quick basic knowledge about the enzymes. All right, here I made a table to kind of summarize the major enzymes in different parts of the digestive system, and those enzymes are more likely to be on TEAS. In the mouth, we have amylase. Again, remember this. I've seen questions about amylase a lot. So amylase is in saliva, right? It's produced by the salivary gland. Amylase digests starch. Starch is a bigger complex carbohydrate. So if you get a question that asks you which of the following, does chemical digestion start, the answer would be in the mouth, in your oral cavity. Stomach, we talked about this a little bit earlier. Stomach has a pepsin, which is the enzyme that digests proteins. So if you have a question asking you the digestion of a protein occurs first in which part of the digestive system, the answer would be stomach. I want to mention, just in case, we all know stomach is very acidic, right? And then there's a reason for that. Now, of course, the acid can kill pathogens, right? Another reason is that the acid can activate pepsin. So the precursor for pepsin is called a pepsinogen. When pepsinogen is in a very acidic environment, it can be converted to the active form, which is a pepsin. So pepsin can digest proteins. And the pepsinogen is created by the chief cells in the stomach. Chief cells. I don't think TEAS will ask you about chief cells and chief cells secreting pepsin, but just in case. All right, next part is small intestine. Now, most of the digestive enzymes are going to be located in the small intestine. So small intestine itself make a brush border enzymes. And why it's called brush border, I will explain in just a second. So these brush border enzymes can break down proteins, can break down lactose, right? When you drink milk, milk has lactose, the milk sugar, that's going to be broken down by lactase. Now, for people who are lactose intolerant, these people do not have lactase. They do not have the enzyme that can break down lactose. And if you don't break down lactose, it's going to sit in your digestive system. It's going to cause all those symptoms like bloating. We also talk about that some of the accessory organs deliver their chemicals into the small intestine to aid digestion, right? So a lot of the enzymes come from the pancreas. So in the pancreatic juice, there's amylase, there's a trypsin, there's a lipase. Amylase, you already know, but trypsin That's a new enzyme. So trypsin breaks down proteins. Very important. And lipids break down lipids. Let's talk about the stomach. So you may notice that the numbers are not really correct because there wasn't a slide on stomach previously when I made the first video, and then I realized, oh no, I didn't have a slide specifically for stomach. So I inserted this slide, so that's gonna kind of mess up the other numbers. So the small intestine was originally number 4, so now it's number 5. Okay, so I just want to point that out. All right, now stomach is connected, it's kind of in between the esophagus and the small intestine, right? So the esophagus is going to have a peristalsis, the smooth muscle contraction to move food down. And then the food is going to enter the gastric sphincter to enter the stomach. So sphincters are usually kind of rings of muscles. So it could be smooth muscle, it could be skeletal muscle. Once the food enters the stomach, the gastric sphincter is going to close. So that's going to prevent food going back into the esophagus. Stomach is the site of first protein digestion. Remember, we talked about the enzyme that's activated in the stomach that performs the protein digestion, which is pepsin, right? Pepsin, that's the active form. The precursor is pepsinogen. Stomach, we all know that is very acidic. So the acid that's produced by the epithelial cells in the stomach is hydrochloric acid, HCl. So that's the acidic, and that's the acid in the stomach. Now, stomach needs a lot of protection, right? When you think about it, because the acid can be very corrosive, and so are the digestive enzymes. So some of the stomach inner lining cells do secrete mucus. So the mucus can protect the stomach from potential damage from the acid and the digestive enzymes. Okay, last thing about the stomach is a term called chyme. Digestion, especially for carbohydrates, for starch, starts in the oral cavity, right, by the amylase in the saliva. And then in the stomach, you have pepsin to digest proteins, and then you have this kind of mixing and churning from the stomach smooth muscle. When you look at the food substance in the stomach, it has been kind of half digested and mixed with the digestive juice, digestive secretions. So that kind of semi-fluid mass is called chyme. All right, so make sure you know what this term refers to. So you will have chyme in the stomach, and the chyme is going to enter the small intestine. That's still kind of that semi-fluid, half digested, half not digested, and food substance mixed with digestive juice. So that kind of mass is called the chyme. Okay, now a few things about small intestine, just in case you see questions on these. Small intestine has 3 sections, right? Remember the order: duodenum, jejunum, and ileum. And again, the main site for digestion is going to be duodenum, right? Because it receives a lot of enzymes, right? And also has a lot of enzymes from its own. Duodenum is connected to the stomach, right? So when the kind of chyme coming from the stomach get down to the duodenum, it's going to be very acidic. Stomach is equipped with a protection mechanism, right? So it's okay to be acidic. But duodenum is different, right? Duodenum does not have all the protection, the mucus, to protect itself from the strong acid. So duodenum is going to receive some chemicals from the accessory organs to kind of neutralize that acid so that your duodenum does not get damaged by the strong acid. The pancreatic juice has a bicarbonate. So bicarbonate is a buffer, is a pH buffer. So that's going to increase the pH and neutralize the acidity. Another thing is coming from the liver. So the liver secretes a bile, remember? Now the bile juice is alkaline or basic. So again, this can neutralize the acidity. Okay, next I want to talk about some of the special structures in the, especially in the small intestine to help digestion and absorption. So the first structure is called villi, right here, villi. And the villi, so think of them as folds in the small intestine. When you look at the inner lining of the small intestine, it can be flat, right? But in reality, the small intestine has a lot of folds. You can see that it's not flat. And these folds are called villi. And when you have these villi, these folds, you can imagine this is going to dramatically increase the surface area, right? Compared to if it was flat. So this is good because the more surface area we have, the more digestive enzymes we can have to digest food, and the more contact area where we can absorb the nutrients, right? So that's why we have these special structures like a villi to increase surface area. Now we have additional structure, more kind of microscopic structure that can further increase the surface area. Now what happens is here, I'm going to use this diagram right here. So we can see these are villi, right? Okay. Now, if we just focus on one of the cells, so let's say we look at this cell right there, okay? So let's say that's the cell we're looking at. So you have, a nucleus. Now, there are two scenarios. First, the cell facing the lumen of the small intestine, facing the empty space of the small intestine, this part can be flat, all right? It can be flat. Or we could have a cell that has this little kind of hair-like structures, kind of like a villi, but much smaller, to increase the surface area. So you can see, I can make this a little bit wider so that you don't think I'm being unfair because this cell is much, much wider. Okay. So there you go. All right. Now, if you have a flat surface as compared to the one with a little protrusion, little extension of the plasma membrane, you can imagine this one is going to have more surface area, right? So these microscopic hair structures, those projections, those are known as the macrovilli. So basically, the macrovilli are at a much, much smaller scale than the villi. Those are just protrusions of the plasma membrane of a single cell. All right, now, again, the villi, the macrovilli, they're there to increase surface area so that we can better absorb nutrients, and it will, provide more ground to have digestive enzymes to digest food. Okay, next structure, liver. So liver is a pretty important organ, so that's why I want to have a separate slide for liver. I've seen not too many questions, just kind of one or two questions on the liver. First, you need to know that liver secretes bile, right? And then bile is temporarily stored in the gallbladder. Okay. The blood that contains all the nutrients will go through the liver so that liver can further process nutrients. Now, that's not very important. I have never seen questions on that. So just know that fact, but you do not need to know any details on that. The next one is important though. Liver does store glucose as glycogen. Now, when you eat food, when you eat carbohydrates, they're digested to glucose, and then all the glucose molecules are absorbed into your body. We often have excessive amount of glucose, and that's when your body will store the glucose for future use, right? And we have a low blood sugar level, that's when you're gonna mobilize the excessive glucose that you have saved before. So how do we store glucose? We store glucose as glycogen. So glycogen is a polysaccharide. So poly means many, and psych, this means sugar. Okay. So think of a glycogen as similar to starch, but it's produced by animals. Okay. Plants, they make starch. They store their glucose in the form of starch. But we don't make starch, right? Humans do not make starch. We get starch from food. So for animals, we don't make starch. Instead, we store glucose in another complex carbohydrate called the glycogen. So both of these are polysaccharide complex carbohydrates. There are just different forms of storage for glucose by plants and animals. Okay, so remember, glucose is stored in glycogen, in the form of glycogen. And once your blood sugar is low, or when you need glucose, your body can break down glycogen to individual glucose molecules. Our liver can also detoxify. For example, when you drink alcohol is actually toxic to your cells. So the liver cells contain enzymes that can break down alcohol. All right. Next thing, I have seen a question on this. So liver performs deamination. What does it mean? Basically, it means it removes the amino group, which is NH2, from amino acids, right? That's just kind of normal metabolism. You break down amino acids. But the problem is when deamination happens, the amino acids is converted to ammonia, and the ammonia is toxic. But no worries, the liver can further change ammonia to urea. Urea is non-toxic. And how do you get rid of urea? It's going to be taken up by kidneys and become part of the urine. Again, urea is a metabolic waste, right? Your body utilizes proteins, and the amino acids are the building blocks for proteins, right? So when you metabolize proteins, metabolize amino acids, there's going to be urea generated in the end as a metabolic waste. So this kind of metabolism is done by liver, right? Convert amino acids to ammonia, but then ammonia is toxic, so ammonia will be further converted to urea, and then you get rid of urea in the urine. Next, we're going to talk about hormones involved in digestion. And I have listed 4 important hormones right here. I'm going to talk about the last 2 first because those are more important. So insulin, you need to know that insulin is secreted by pancreas. Very important. Pancreas. So insulin is a very important hormone that regulates the uptake of glucose by cells. Now, glucose is the universal cell fuel. Okay, so this is how cell can obtain energy to survive. Glucose comes from your food, right? So remember, after the carbohydrates are broken down to glucose, glucose will be absorbed in your GI tract and enter your bloodstream, right? And then the bloodstream can deliver glucose. Just do G, glucose molecules to all the cells. There we go. All right. So let's say this is a cell, and the cell needs to have glucose for energy. First, the cell has to be able to take in glucose molecules. Okay? So the glucose has to be inside the cell. And then the cell can use mitochondria to metabolize the glucose, break down glucose to extract the energy, and then store the energy in the form of ATP. Insulin is critical to this process, right? Because without insulin, so this is with insulin, and you can see glucose can enter the cell. Now, without insulin, the glucose cannot go into the cell. So the cell basically does not have any fuel, right? Does not have energy source. This is kind of like your car without gasoline. A car can't do anything. Right? So that's a problem that people with diabetes face. Their cells cannot take in glucose for energy source. So usually their cells will utilize other molecules such as proteins to get energy. Not ideal. Okay, that's insulin, and make sure you know that insulin is a hormone. It's secreted by pancreas. Next hormone is a glucagon. So glucagon almost does the opposite. So the glucagon will increase will increase blood sugar level. I forgot to mention that insulin decreases blood sugar level because the glucose is taken in by your cells, right? So their level in the blood will drop. Now, glucagon does the opposite. Glucagon is going to increase your blood sugar level by stimulating the breakdown of glycogen in the liver. So we mentioned this, usually glucose is stored in the liver in the form of glycogen, right? Glycogen is a polysaccharide, and it's really kind of polymer of glucose. My drawing is not very accurate. So this is these are individual glucose molecules, right? Okay, so you can see they're all linked, right, to form a very big molecule. And this entire molecule is called the glycogen. When you have a low blood sugar level, then glucagon will be released, and the glucagon is going to stimulate the breakdown of glycogen into individual glucose molecules, and those glucose molecules will enter your blood, and that will increase the blood sugar level. Okay, all right, that's insulin and the glucagon. The next two, leptin and ghrelin. So leptin lead to satiation. Basically, leptin will give you the feeling of being full, like you have had enough food. So that's what leptin does. Ghrelin does the opposite. It induces hunger. So ghrelin is produced in the stomach, and when ghrelin is produced, that's when you feel a little bit hungry and you want food. Okay, so those are the hormones involved in digestion. Now let's look at some practice problems. Again, if you need some time to think about the question, just pause the video. Okay. All right, number one, breakdown of which of the following begins in the stomach. So the correct answer is protein, right? Because stomach has an enzyme called a pepsin that can break down proteins. There are any enzymes that can break down proteins in the mouth, right? They can break down carbohydrates, right, which is specifically starch. Number two, which of the following produces ammonia by deamination in the liver? So the correct answer is A, proteins. Remember, proteins will be broken down to amino acids, right? And then the liver remove the amino group from the amino acids, which will become ammonia. So proteins. So ammonia, urea, those are metabolic wastes of proteins. Number three, which of the following tissues is responsible for contractions during peristalsis in the digestive tract? So the correct answer is smooth muscle, right? So peristalsis is not a very forceful kind of movement as the movement generated by your skeletal muscle. So skeletal muscles are a lot stronger and their contractions are stronger. But in the digestive tract or in your urinary tract, you don't want that much force, right? That will be very painful. So you want some kind of slow, more kind of mild muscle movement. So that's generated by smooth muscle. Number four, type 1 diabetes is a disease caused by the lack or low level of which of the following hormones? Diabetes, that's going to be related to insulin, right? Wow, good job, guys. You have completed the digestive system. Again, if the video is helpful, let me know by subscribing or leaving me a comment, especially if you want me to talk about specific topics or if you have any questions about any of the content in the video. Feel free to let me know. I will try to respond to your comments as fast as I can. All right, guys.